provided with urine samples, and each urine sample would have been tested with a strip. So here's a bottle that's the strips come in. Here are the strips. Strips are very easy to use. All we do is dip the strips in urine, count 10 seconds, take them out, set them on the counter for about 20 seconds or so, and then we'll read the color changes, okay? So all the little squares you see here are associated with different tests. So one test strip can test for several different urine contents, okay? So for example, this strip will test, this one here will test for blood, white blood cells, leukocytes. Will test for proteins and glucose and ketones and, all sorts of other things. So the idea here is that you are going to be given essentially what amounts to a key, which is what this is. This key here indicates the levels of the various things tested for based on the colors that your strip changes to, right? So this here is showing you normal as test strip, right? So it's like this person is holding a test strip up against the chart over here, okay? So this test strip has little squares on them. These little squares here are associated. So for example, this one here, square here, that square is for glucose. So this first square at the bottom is for glucose. And the color here is what we associate with our key over here. So for example, this color, these two colors look about the same. So we'll say that this strip here would come back as negative, right? Because they have the same color. If the test strip had come back and it was a brown color like this, we would say it contains well, if you look here, so there's a number. Oops. Image is a little blurry, but you can still read it. This is 30, right? So this is saying 30 millimoles per liter, that's right. So this is basically saying for every liter of urine, you would have 30 millimoles of glucose in it, which is pretty high, right? So what you would do is you would now go down to the strips provided. I think there's A, B, C, D. Oops. Wow, that's tiny. You would look on your strip, right? And you'd compare it to your chart. So you know what I normally do is like a thing like this, where I just take the chart and I'll stick it in like PowerPoint or something. As I said, I find this is the easiest way to do it. So stick the chart in PowerPoint. And then I just come back and then I stick the I just copy one of these. So that's A. And just paste it in right beside it. Now the trick here is to ensure that they're approximately the same height, so that you can line up your colors, right? And once you have them lined up, we look here, that's a green, right? Let's just do the slideshow for this. One second. Okay, let's try that again. So here we have our strip. Let's do slideshow for this one. All right, so I will look and say, okay, here's a green strip. That's what glucose, which one of these greens does that glucose look most like? I said this one, so that means glucose is negative. Then I look at bilirubin, and I said, boy, do any of these look like bilirubin? No. The negative one looks closest to it, so I'll say that's negative. Ketones, I look at the color for the ketones here. And I look at this one here, it seems closest to negative. So I'll say ketones are negative as well. Then specific gravity, this is looking at how much heavier the urine is than water. The heavier it is, the more dissolved substances are in it. So I say, okay, this one looks kind of brownish. Then I look around until I see the brown it looks most like. And I'm going to say this one, right? So that's like 1.025. So that gives me the specific gravity of this urine. Then I check for blood. This one looks kind of brown too, which means it's not negative. This color here looks most closely like that one here. And that says 25. So that means a small amount of blood is in the urine. For pH, once again, here's the color I have here. And I look for the matching color. It's about 6.5 again, right? So that's the basics of what you're going to be doing. As you go along, each square, sorry, you're comparing it to get a reading. Then what you're going to do is we're going to put these readings in the table. So here's the table for each urine. You indicate either negative or positive, negative, depending on what, or positive, depending on what your strips say. And if your strip is positive, you're going to write the value of the positive. Remember, the value is here in the chart under the color or above the color, depending on which one, right? Above the color. And once you fill out the table, you are going to assess the urine. By that, you'll indicate whether the urine is normal or abnormal. And then you'll indicate which tests it's abnormal for. So if, for example, if it's high in glucose, you're right, abnormal urine, glucose high, protein high, whatever, right? So that's what you look for A, B, C, D. Then now, here we have a little question here associated with a case study. It's very simple, very straightforward. This is actually probably shorter than the one in your lab manual because you're not doing any of the actual experiments, right? So this should be easier for you. And you don't need me to talk you through it. But I will upload this lecture to the YouTube channel just to ensure that you have this part of it. Okay? All right, any questions so far? All right. Awesome. So let's move on. It's 6:20 now. And I see we're just at 39 participants of the 60 odd we're supposed to have, but such is life. All right, so let's continue. Go ahead.
Good evening. There's a lymphatic tutorial to do.
There's a lymphatic tutorial to review. As you would have noticed that there's no turn-in for that tutorial, right?
All right, sir. I think there's two tutorials on cardiovascular, sir. Could you open
There's
back
one
the
on
second
blood
one,
and
please?
one on cardiovascular.
Oh, sir, could you open? I
Yeah, I'm
think
not
it's
reopening those.
cardiovascular.
No, I'm not reopening them. If you haven't submitted it by now, your time gone. That's way too long, man. Come on, this is ridiculous. They've been here for weeks. All right? I've moved on with my life. There are two open slots there in the tutorials for the respiratory anatomy and the respiratory physiology ones. All right? Let me briefly run to that. And then we can start our class. But yeah, I don't think I should be opening a tutorial after 3 weeks. That's ridiculous. All right. Right. So down in the respiratory section, you're going to see turn-ins once again for Thursday and Friday of this week, of course, because it's due this week. We have respiratory anatomy and respiratory physiology, right? Respiratory anatomy for tomorrow, and you have to submit it by 4 p.m., just before the class starts. For the Friday group, you have to submit the anatomy one by 2 p.m., right? That is tomorrow. And sorry, Thursday, Friday. You also have the physiology one to submit, same dates, same time. So you're turning in 2 tutorials tomorrow, one anatomy, one physiology. I will be putting up a urinary system tutorial later this week as well. Y'all need to ensure that you go through the respiratory system tutorials. You're never sure of what might show up on the test that comes from the tutorials, right? And if you do not submit tutorials on time, you will submit them late and they will not count towards any bonus. Y'all taking things for joke, man. Right?
Sir.
Hello.
You're saying if it's a case, so we don't register yet. How we go submit them?
If you are from Mobi, I understand why you're not registered. If you're not from Mobi, I don't understand why you're not registered.
Sir, I don't understand either.
Because they opened up additional spaces Friday. When was the last time you tried to register?
Sir, last week Thursday after tutorial class.
Well, there you have it. Try and register again today. Because they said that they opened up spaces last week Friday. Right? They should have opened 10 spaces last week Friday. All right? If you actually are still having issues with registration with regards to, send me an email, I'll ask about it, right? But it's like week 5 now, man. That's ridiculous. Anyway, it's not your fault, it's not your fault. I can't do anything about it. I'll figure something where you're concerned. Otherwise, if you all have been registered, then my tutorial should have turned in. The thing open for a long time. Anyway, we'll leave it alone. So please remember, we have 2 tutorials to submit this week. It is in your best interest to do them before the test tomorrow. I will leave it at that. You decide what you want to do. Yeah.
You're saying tomorrow for the physiology, but I'm seeing the 20th. Isn't that Thursday? And not tomorrow.
Is the in-course test. That's what I've been talking about. The tutorials are due on Thursday, Friday, which is a different thing. Okay. All right. Where are we now? Yeah. So let's continue. See if we can finish urinary system today. Because that will definitely be a big player in your next in-course test. It is now after 6:25. I have 44 people here. So I'll make my last announcement here about the test. Anybody ask me after this, I will not respond. Because they should have been here. Test 1 tomorrow, which is the 19th of June, 47C1 at 11 a.m. Go ahead, Hannah.
Sir, any updates on what will happen to the Montego Bay students?
I haven't the foggiest idea. What I indicated was that I might, because of all the confusion with the Monday thing, I'm actually going to have to set a separate test for all of you, which is a great aggravation, but I can't help it. That is nothing else I can do. So I'm trying to arrange for it to come down next week. All right. So maybe that's for the best.
Okay.
I don't know, but it's not your fault. Once again, if it's not your fault, then it is for me to figure something out, right? So I'm going to redo it tomorrow. I will set another test and I will try my best. Hopefully there's somewhere, but to carry it down Thursday or Friday the latest, right? Hopefully. But if you have the Friday tutorial, ask me on Friday what happened, right? All
Okay,
right.
sir.
Yeah, man. As I said, I'm trying, but sometimes either you take a get a fight. Anyway, it is what it is. So once again, for Papine people, test 1, 47C1. Y'all should know where that is by now. Does anybody not know where the 47 buildings are? All right, good. So y'all know, I think it's still, I think C is the 3rd floor, so that's the top floor. So 47C1, 11 o'clock. As I said, starting latest 11:05. If you come after that, you will not be given additional time. If you come after 30 minutes, you will be required to do the supplemental, which is going to be all essay questions, and you will have to find the time to do that when I have the time to do that, not when you have the time to do that. I'm dead serious. I'm not going to go out of my way if I miss a test. All right? More likely than not, it will be on Monday. No, all right. Let's not say Monday. I have an hour in between the 2 labs, which is normal lunch, but I guess I can skip lunch. I got enough fat I can manage. And you'll do it in the office. But it will be essay only. I'm telling you from now. I can't be rewriting so much multiple choice questions. Go ahead, Alicia.
Sir, what you said about the test tomorrow, I went offline
Oh, Jesus.
from Monday.
You see my screen, right?
Yeah. No, for
Cool.
mobile students, you said you're going
Oh,
to
for
set a test
mobile
for them.
students. Okay.
Yes. You
Okay.
can,
Okay, look, okay.
uh,
As I'm saying, I'm trying to arrange for y'all to do the test next week because the bus down didn't work out in time for this week. All right. I will update everybody Friday. I did tutorial, right?
okay. So,
Yeah.
sir, when is the second test?
It's gonna be the last week. And I will have hopefully we'll have sorted out our system by then, so we won't have this problem for that test.
Last week.
Yeah. So what I mean, normally, the first time for anything is always rough, right? So the second time things should be smoother. Right? The final exam, I don't know what's going on with that. You may still have to come up for that one. I haven't really heard any accommodations being made in Mobile for the final exam. You might have to check in on that one, since I'm not in Mobile. But you can let me know, right?
Sir,
Yes.
yes. I just wanted to know, being as you said you're going to set another test for the MOBI students, would it be multiple choice or essay type questions?
I am not going to penalize you for something that is not your fault. Y'all should know that by now. So you're not late. You're not absent. You're going to get a regular test. Right?
All right, sir, thanks.
Which means I'm setting 3 tests. What such is life? Right?
Sir,
Uh-huh.
about the tutorials, what should we do about that?
Once again, if it's mobile people I'm talking to, it's not your fault, it's UTEC's fault. You will not be penalized for it, right? Have no fear, we'll figure something out.
Oh, all right, sir. Just one last question. When you say the last week, is the last week of this month or next month
Last week of
for the
TK,
test two?
which is basically, what's that, 4 weeks from now, I think?
Alicia, you're in the group. Information is in the group. Anything wants to say, just ask in the group. Make the man teach him class now.
Thank you,
No,
dear.
no. Me no see. Going like, all right, then fine.
All right, let's move on then. So remember, 47C1 tomorrow, 11 a.m. for people who are in Kingston. If you do not show up, for whatever reason, you cannot get the same test as everybody else. Obviously, you will be getting an essay-based test. I'm telling you from now, I'm not rewriting a test for you. Mobi students, I will send you a separate test for next week. All right.
Sir,
Now,
we're getting lab quiz tomorrow. Sorry to interrupt.
absolutely, lab quizzes are based on respiratory system, so you should have studied for them. So this is the
Okay.
best time to give it, right? With everything fresh in your mind. As a matter of fact, the 8 o'clock group, you will be getting a lab quiz before you come to the test, which means at the very least, the lab quiz should help you to gauge. Well, I really hope everything work out tomorrow. Because if you mash up the lab quiz, you know so you're ready for the in-course test, right? But we'll see what happens. All right. Filtration. Let's talk about filtration. What we're trying to do here is to explain how the blood pressure is going to affect the ability of your glomerulus to filter substances from your blood, okay? Now, this is also related, generally speaking, to blood pressure, okay? And what determines blood pressure. So you'll notice here that we have some terms, okay? So we're going to need to, of course, learn these terms. So for example, here we have this lovely one here called blood hydrostatic pressure, BHP. Generally speaking, I will write out the terms. So if I'm doing a question, I usually spell out the terms so that I will not say, so if I will have blood hydrostatic pressure, and then usually in brackets, I'll put BHP. But let's say, for example, that you have to do a lab quiz and you have to write an answer, or you do, right? You don't have to write out blood hydrostatic pressure. All you have to write is BHP, okay? But actually, I think the respiration lab quizzes are all MCQ, if I remember correctly. It's probably a moot point. Anyway, so what is blood hydrostatic pressure? Fundamentally, blood hydrostatic pressure is the force generated by your heart into your circulatory system. So when your ventricles contract, your ventricles push blood into your arteries, right? Now, I'm assuming everybody remembers that blood is incompressible, right? By that I mean you can't squeeze. And the reason why blood is incompressible, by the way, is because of the water content, right? Water cannot be compressed. You can't squeeze water into a smaller shape or a smaller size, right? This is one of the reasons, of course, why let's say somebody jumps off of something high and jumps into water. If they're belly flopping it, it's almost like hitting concrete. Because when you hit the water doesn't flex under your body. The water just shoves all the force right back up at you, slams the force into you, mash up, and then it slowly moves away from you to let you sink in, right? So when your heart contracts and it pushes on the blood and forces the blood out of itself, all the other blood in the circulatory system also gets a shove, right? And that shoving force from the heart is what we call blood hydrostatic pressure, okay? Hopefully that makes sense to you, right? So when the stronger your heart contracts, the stronger your BHP is going to be. Oops. So, of course, I want you to remember now, this is your afferent arteriole. So AA for that one. This one is your efferent arteriole, EA for that one, right? Efferent arteriole, afferent arteriole. As usual, you'll notice the afferent arteriole is wider than efferent. We spoke about this already. So So the force of the blood shoving in, right, will mean the BHP is relatively high, because a large volume of blood is going in because of the wider tube. The same volume of blood has to go out, even though the tube is narrower, which means that the pressure inside here has to increase, okay? But as the pressure increases, because of the fenestrated capillaries, substances filter out of the blood and into the Bowman's capsule, right? So the BHP is a force pushing things out of the blood when it's in the glomerulus, okay? Now, at the same time, you have forces trying to push or trying to hold substances into the blood. One of them is colloidal osmotic pressure. Another one is capsular pressure, right? So colloidal osmotic pressure is basically what pretty much it comes from the blood itself, right? So the proteins in the blood want to hold on to the plasma and other substances in the blood. So colloidal osmotic pressure is coming from the substances already dissolved in the blood. So they like to keep all the nutrients and everything in the blood around them. They love the stuff. So albumin, for example, which is a major blood protein, it doesn't want you to squeeze out the water, doesn't want you to squeeze out the glucose. It's trying to hold on to all of these things, right? Which means now, in order for you to squeeze these things out, your blood hydrostatic pressure has to be high enough to overcome this colloidal osmotic pressure that is trying to hold on to everything, okay? So the BHP is trying to force things out of the blood. At the same time, the COP is trying to hold on to them, right? And in addition to that, you also have capsular pressure. So the pressure inside the Bowman's capsule itself. So as the Bowman's capsule starts to get filled with filtrate, it is going to resist more filtrate coming in. It's there already. It doesn't want to make space for anybody else, basically, right? So that also resists substances leaving the blood. So we end up with two forces trying to push things out of the, a major force trying to push substances out of the blood, and two smaller forces trying to keep substances in the blood, right? So in order to determine the overall force pushing out, we have to subtract, right? Which is why, of course, down here we have the BHP, which is on average about 60 millimeters of mercury. That is trying to force the blood out. The COP around minus 32, which is, remember, that is coming from the blood itself, substances in the blood trying to keep themselves in the blood. And that's minus 32. And then the capsular pressure, right? The Bowman's capsule itself not wanting any extra stuff from the blood, trying to push it back in. When we subtract the negative forces from the positive forces, generally speaking, on average, we get about 10. So the force trying to make the filtrate, the effectively, which is the net filtration pressure, is going to be about 10 millimeters of mercury. Okay? Remember, this is average. So as long as we have this amount of net filtration pressure, it means our kidneys will function properly. Right? So all is well, you're making urine. There's enough force to push substances out of the blood and into the Bowman's capsule, but the force is not so great as to damage the glomerulus, right? Now, obviously, at least I hope it's obvious to you, if let's say, for example, your blood pressure drops, right? Your blood pressure drops, let's say you have, for example, but well, lots of things can happen to you. Let's say, for example, you're dehydrated. Let's do the easy one. You're dehydrated. You haven't drunk water in a long time. You know, you've gone 2 days without water. Of course, this means that the volume of blood you have is going to fall, right? And if there's less blood flowing to the system, as your heart is pushing out the blood, there's a lot more space for the blood as it pushes it out. So the pressure on the system drops overall. That now means that when the blood flows into the capillaries here, the pressure squeezing fluid out of the blood is going to be lower, right? So instead of a BHP of 60, it might be 55. So you might have 55 here instead of 60 if the person is severely dehydrated, which might mean you end up now with the overall net filtration pressure of 5, right? Which means that you're making urine, but you're making a lot less urine than normal, which of course is a sign of dehydration, right? On the other hand, let's say you drink a large volume of water. So you've now increased the volume of your blood because of all the water you've added to the blood. So when the heart is contracting, you're forcing a bigger volume of blood out, which means more pressure. BHP goes up. All of a sudden, the net filtration pressure also goes up, which means you're making urine even faster than normal, which of course, as you know, when you drink a lot of water, you make urine pretty fast. Okay? So I'm just trying to link these things together, right? Okay. Now, your glomerular filtration rate is essentially looking at the filtrate formed per minute. Okay? So every minute the kidneys function, how much filtrate is squeezed out of the blood. Now, to determine your glomerular filtration rate, you need to know something called the filtration coefficient, which has a standardized value. So if I ever ask you to calculate glomerular filtration rate, I will always give you the coefficient, right? Now, all you need to do is multiply the coefficient by the net filtration pressure. And we just spoke about how to get net filtration pressure here. Remember, blood hydrostatic pressure minus colloidal osmotic pressure minus capsule pressure. All right? That gives us our net filtration pressure. So once we get the NFP, we multiply it by the coefficient, which remember, if I give you a question, I will give you the coefficient for sure. That for on average is around 125 mils per minute. So that means that every minute of the day, realistically, it's faster during the day and a little slower at night. Well, I should say it's faster when you're awake and slower when you're asleep. You're generating about 125 mils worth of filtrate, which adds up to 180 liters of filtrate per day. Now, I want you, at least I'm hoping you realize that you're only going to have 4 to 5 liters of blood. If you're a male, all right, well, let's just call it an average about 4 to 5 liters of blood, right? So if you have 4 to 5 liters of blood in your body, how are you producing 180 liters of filtrate? Simple. The vast majority of that filtrate, 99% of it, has to go right back in the blood where it came from. So you're constantly cycling filtrate from the blood through the kidneys back to the blood, constantly cycling it round and round and round, right? And you're only losing between 1 and 2 liters of urine per day. Now, keep in mind, 2 liters is on the high end, right? I mean, how many of us are going to produce a Coca-Cola with a 2-liter soda worth of a urine a day? Especially when it's this hot, you're probably doing more like 750 mils, or somewhere in the 1 liter range these days, right? All right. No questions yet? Let's keep going. Now, if glomerular filtration rate increases, obviously urine output is going to go up as well, right? And the more urine you produce, of course, the more water you will lose. Therefore, you will get dehydrated faster, right? And keep in mind, and why does that lead to electrolyte depletion? Because as you're filtering out to make the filtrate, it's not just water alone you're losing. You're losing salts, various minerals, etc., etc. All of these things are electrolytes, right? And no matter how clear your urine is, there will always be some amount of salts in it, some amount of electrolytes. So the more urine you produce on a daily basis, the more electrolytes you lose. So if you produce a lot of urine, you will become electrolytically depleted, right? I could also say to you that if you sweat a lot, especially the way the times are hot, you will also end up depleting your electrolyte levels in your blood, and simply drinking water is not going to replace that, right? So it's weird to say, but when the time is very, very hot, you need to eat salty food and not just drink water alone. Because at least you get back the sodium and the chloride from the salty food to help to add to your electrolytes that you're losing from the sweat, right? Anyway, so buy a bottle of water, yes, absolutely. But maybe have a banana chips too, you know? Anyway, if your glomerular filtration rate falls, of course, it is going to be harder to make the filtrate. Therefore, less substances get filtered out of the blood. And the main thing you're trying to get rid of course, which of course is your nitrogenous waste. You're not getting it out of the blood fast enough, so you develop azotemia. Remember, azotemia is higher than normal nitrogenous waste in your blood. And if the azotemia gets bad enough, just to remind you, it leads to uremia. Right? And uremia now means that your the nitrogenous waste levels in your blood are so high that you're actually starting to get sick. Right? So azotemia, your nitrogenous waste levels in the blood are above normal. Uremia, the levels are so high, they're starting to make you sick, right? Now, glomerular filtration rate is going to be controlled by, of course, adjusting the pressure in the glomerulus itself, right? So if you want to increase filtration, you increase the pressure in the glomerulus. If you want to decrease filtration, you decrease the pressure in the glomerulus, right? Now, obviously, this is not something you have to think about on a daily basis because, of course, it is controlled via an auto-regulatory mechanism, right? So that is autonomic control. And it is controlled by a sympathetic nervous system. And it is directly impacted by hormones like renin and angiotensin, which you're going to explain how those work, okay? So your urine production is controlled by your nervous system with inputs from your hormonal system, right? Both work together to regulate your urine output, right? Okay. Now, what I have here is called the juxtaglomerular apparatus. This thing here is very, very important when it comes to that auto-regulation I just spoke about, okay? Now, just to remind you all, juxta, as in juxtapose. I know you might have heard juxtapose in literature class. I never actually seen it used anywhere else. At least that's the way it was for me. But juxtapose, remember what we said? It means you're putting two things side by side, essentially. All right? So so juxtaposing something, glomerular, so it must have something to do with the glomerulus apparatus. So we're juxtaposing the glomerulus with something. We're putting the glomerulus side by side with something. And the thing we're putting it side by side with is your distal convoluted tubule down here. All right? So the juxtaglomerular apparatus is made up of the renal corpuscle here, which contains the glomerulus, of course, the afferent and efferent arterioles, because, of course, they go into and out of the glomerulus, as well as your distal convoluted tubule. Right? There are cells in here that are going to constantly measure the concentration of your blood, right? And determine whether or not there's too much water, too little water, and then help to determine, do you need to filter out more or filter out less to preserve the water you have? So your juxtaglomerular apparatus is going to control, at least in a greater part, the filtration and hence urine production, right? All right. Obviously, being able to label this is important, okay? Lab quiz is coming up soon. You're going to need to be able to label this, right? So, we have here, within the distal convoluted tubule, we have a specialized set of cells here that make up something called the macula densa. And the macula, and I'm going to talk about it more in a little bit, but the macula densa here is monitoring the salinity of the blood. If the blood gets very salty, it means that we need more water in the blood to dilute the salt. So, of course, that means that we need to reduce urine production, if that makes sense to you, right? And, of course, if the blood is not salty enough, there's too much water in the blood normally, of course. So you increase urine production, right? And over here now, we have the afferent arteriole, and we have vasomotion. As you know, vaso is referring to the blood vessel, and motion, of course, is movement. So this is going to cause, essentially, the afferent arteriole to constrict or dilate, right? And we're going to explain how that is going to help either form more urine or form less urine, okay? All right. So far, so good, I hope. Let's keep going. So see if this makes sense to you now. As your blood pressure rises, right, you're going to have a constriction of the afferent arteriole and a dilation of the efferent arteriole. And as blood pressure falls, you have the dilation of the afferent arteriole and constriction of the efferent arteriole. Now, let's explain why this is happening. Let me do a little diagram for this one. Real quick. Let's do a little diagram real quick. And then you tell me if it makes sense, right? So remember now, as your blood pressure rises, you're restricting the flow of blood into your glomerulus. So here we have our afferent arteriole, right? That dips down, of course. So afferent arteriole glomerulus, right? And of course, we have our Bowman's capsule around it. Now, See if this makes sense. If the blood pressure going into the glomerulus is very, very high, right? It will be high enough to actually overcome the podocytes. Remember, the podocytes are wrapped around the glomerular capillaries, trying to keep them together. If your blood pressure is extremely high, the podocytes just can't, they don't stand a chance. Your blood vessels will just start popping like overfilled balloons, okay? Which means, of course, that your kidneys will take damage. So if your blood pressure goes up, you can damage your kidneys, right? To prevent this from happening, okay? As your blood pressure goes up, you're going to constrict the afferent arterioles. So you narrow them down and you dilate the efferent ones, right? So what does that do? It means that you're restricting the flow of blood into the glomerulus because you've now narrowed the tube in. And by opening the tube out, it makes it easier for the blood that is going in to come out. So the blood flow in is easier. Sorry, the blood flowing in is reduced. And you make it easier for the blood flowing out to leave. So the pressure in here will drop. So far, so good. Makes sense, right? Now, if your blood pressure is too low on the other hand, so that it's so low, net filtration pressure starts to fall. So your kidneys are no longer functioning properly. So what do? You will dilate now. You're basically doing the opposite. You're going to dilate the afferent arteriole here, and you're going to constrict the efferent arteriole. Why do you do this? When you dilate the afferent arteriole, more of the lower pressure blood can flow into the glomerulus. So the blood flow to the kidneys increases. And by constricting the efferent arteriole, once again, it's harder for the blood to leave, which means that there's a buildup of pressure in the glomerulus. So the filtration pressure will rise. Okay? So the ability of your kidneys to control the constriction and dilation of the afferent and efferent arteriole allows us to stabilize net filtration pressure. All right. So far, so good. No questions? Okay. As it says here, you can maintain a stable net filtration pressure for blood pressure ranges between 80 and 170 millimeters of mercury. What does this mean? It means that if your blood pressure drops below 80, your kidneys start to fail because you cannot maintain a high enough pressure within your kidneys to filter the blood. So if your blood pressure is below 80, you will start to develop azotemia, right? You can't filter out the nitrogenous wastes. They build up in the blood, you now have a problem, right? If your blood pressure is over, and by the way, this is the systolic pressure, right? I hope you all realize that. This is systolic pressure. Remember systolic pressure, the pressure you get when your ventricles contract and force blood into your arteries, right? If your blood pressure is above 170, remember normal is about 120, right? Let's just remind everybody about that. Normal blood pressure is 120, that's systolic, over 80. That's diastolic, right? So when you go above 170 now, the pressure is so high that even though you're constricting the afferent arteriole and dilating the efferent arteriole, the blood going into the glomerulus is still under such high pressure that you're going to start to destroy the glomerulus. So at this point now, you're actually physically causing permanent damage to your kidneys, right? Now, technically speaking, periods of high blood pressure are therefore worse than periods of low blood pressure. Because if your blood pressure is low, yes, you will develop azotemia, and you can take medication to increase your blood pressure, and you can take blood transfusions to increase your blood pressure, and then you will go back to normal. So there are mechanisms by which you can raise your blood pressure. It's far more difficult to lower your blood pressure. And usually by the time people realize that their blood pressure is very high, their kidneys have already sustained damage. Right? Their kidneys have already been damaged, and the damage is going to be permanent. Now, I keep telling people this, so I'm sure I've mentioned it before. Your organs do not heal. Okay? The only organ in your body that can regenerate itself is your liver. And by regenerate, I mean make it like new again. The liver gets damaged, it can actually regenerate and make itself new. And it is also the heart, therefore, is the hardest organ in the body to serious damage to, permanent damage anyway, right? All the other organs in your body, if you damage them, the best you can hope for is that they seal off the damaged area, but that area will never function again. Same thing with the lungs. Remember I said the emphysema is permanent, right? Once you damage those alveoli, they ain't coming back. So when you yourself kidney damage, it's permanent damage. You're never going to fix it. You will simply now be living with kidneys with reduced function for the rest of your life. Simple as that, right? Okay. So the control of your glomerular filtration rate is based on negative feedback. Negative feedback, of course, you should have heard of this from when you were doing hormones at CSEC, right? The idea here is that a change occurs in the body, right? Remember that within the body, we have what are called set points. A set point is basically a level to which a physical characteristic of the body is said to be at its optimal, and the body wants to keep it that way, right? So a set point is as close to an optimal point as your body has. So for example, set point for body temperature, 36.5 to up to 37.5, right? Somewhere within there is your best body temperature. Your blood pH has a set point. As you know, 7.35 to 7.4, somewhere within there is the optimal pH for your blood. Once you move outside of the set point, you're going to start causing issues. You're going to reduce the efficiency of your blood, of your body, I should say, not your blood. Things become more difficult, right? So, of course, if your GFR is too low, as we said, your kidneys will not filter properly. If the GFR is too high, you'll not only produce excessive urine, but you'll damage the kidneys, okay? So negative feedback now is designed to oppose changes. So something changes beyond a set point, your body takes steps to put things back to where they should be. All right, so here we have high GFR. So too much urine is being produced. So you have rapid flow of filtrate in the renal tubes, right? So that means now the filtrate, there's so much of it just rushing through the nephrons, okay? It's like a hose under high pressure. It's just swishing through. And because the filtrate is moving so quickly through the nephrons, it is hard to reabsorb substances from it. Remember, you're supposed to reabsorb all of the glucose, all of the amino acids from the PCT, right? Now, this is only possible if the filtrate is moving slow enough, right, for the PCT cells to pull out what they need to pull out from the urine. If the urine is, or the filtrate, sorry, is moving quickly through the PCT can't grab everything out of it fast enough, which of course now means that you're producing urine that contains substances that your body needs. So you will start to have, so when your GFR is very high, you'll start having protein in your urine. You'll start having glucose in your urine, right? More hormones will appear in your urine. Things that should be in your body will be lost, which is bad, right? Now it mentions here that you will have less reabsorption occurring, and the macula densa now is going to sense the high salt concentration. Remember, sodium chloride is also reabsorbed by the nephrons. So if you're not able to pull out the salt out of the urine, your urine will be very, very salty, right? Remember where the macula densa is, it is in the DCT. So once the urine passes through the loop of Henle, it is not supposed to be super, super salty. But if it is, the macula densa picks it up. And then the macula densa now releases a paracrine secretion. A paracrine substance is like a hormone. It's not quite a hormone, but it's pretty close. When we do hormones, we'll talk more about them. We're not sure exactly which one it is, but whatever is released by the macula densa now causes the constriction of the afferent arteriole, right? And the dilation of the efferent arteriole. That, of course, reduces the pressure going into your nephron, and therefore you produce less filtrate, right? Hence, negative feedback. Rising GFR causes all of these things, which lead to a fall in GFR, okay? All right, so far so good. No questions? All right, let's continue. Okay. I mentioned already that the sympathetic nervous system is going to be the one in control of this, okay? Now, you all know the sympathetic nervous system is pretty much there to increase activity pretty much everywhere in the body except in the digestive system, right? So as it says here, strenuous exercise or acute conditions like shock will stimulate afferent arterioles to constrict. So when you're exercising a lot or you're going to shock, you have a constriction of the afferent arterioles. When that happens, of course, your glomerular filtration rate will fall and you will produce less urine. Now, if you think about it, if you're exercising heavily, you're going to need that water that's in the blood, right? Because you'll be sweating. And as you're sweating, you're losing water. So the last thing you want to be doing is producing a lot of urine. So by constricting the afferent arteriole and dilating the efferent arteriole, you make less urine and you keep more water in the body, right? So you're preserving water that way, right? It also mentions here that you're going to also redirect blood to the heart, the brain, and the skeletal muscles. And of course, if you're exercising or you're under fight or flight conditions, those are the places you want the blood. The last thing you need to be doing when you're running away from a gunman is making urine, right? Now, what we're looking at here now is the hormonal part of it. We're looking at what is called the renin I always misspell renin. Okay, renin, angiotensin, aldosterone system. The renin angiotensin aldosterone system, RAAS. Let's pretend that says race, okay? Now, this system is going to be pivotal in controlling several factors related to the reabsorption of water and the preservation of water in the blood. All right. So obviously, you're going to need to learn this one. All right. So this lovely flow chart here, you're going to have to internalize this one, right? That said, I do have it. Where is it? I have it further along, so I'm just going to run through here with it, okay? So this is the renin-angiotensin-aldosterone system in words. So as it says, decrease in blood pressure causes a decrease in the amount of extracellular fluid. That makes sense, right? Your blood pressure drops. It now means that your blood needs more fluid. If there's more fluid around your tissues, that fluid flows into your blood. So your extracellular fluid concentration drops, okay? And decreasing this extracellular pressure near the distal convoluted tubule causes the juxtaglomerular cells to release renin. So what this is saying is that as your tissues around the kidney are losing your extracellular fluid, the pressure around the kidney is going to drop, right? The pressure inside the kidney is also going to drop because of the loss of this extracellular fluid around the nephron. As the nephron is put under lower pressure because of loss of fluid, the juxtaglomerular cells from the juxtaglomerular apparatus are going to release renin. Okay? So renin is now released. You know, let's just go back to the chart. Maybe it'll be easier if you can see a visual of it. You can always read through that later. Let's just use our chart, right? Maybe it'll be easier this way. So here we go. So remember now, you are losing water, okay? You're sweating it out, you're vomiting it out, whatever. The volume of water in your body is starting to fall. Therefore, the volume of water in your blood starts to fall as well. When that happens now, your blood starts to pull water from your tissues. So your tissues now start to lose water. They become more and more dehydrated, okay? They're losing tissue fluid. That tissue fluid is flowing back to the blood to become plasma to maintain your blood pressure. When this happens to the tissues around the nephrons, that now means that, once again, the pressure around the nephrons will fall. And the juxtaglomerular apparatus will pick this up and it will release renin into the blood. Okay? Once the renin is released into the blood, that renin is going to now find angiotensinogen. Remember, when you say nogen at the end of a word, it normally means it's the inactive form of whatever substance you're looking at, right? You remember fibrinogen, right? So the renin now is going to activate angiotensinogen by cutting off the angiotensin from the rest of the molecule, forming angiotensin 1, which is why you go from 453 amino acids to 10 amino acids. It's basically like think of the angiotensinogen as a case that holds the angiotensin 1. Renin opens the case and pulls out the angiotensin 1 and lets it go, okay? So now you have separated out angiotensin 1, which of course is much shorter because it was inside the case, right? Now the angiotensin 1 now is going to be exposed to an enzyme called ACE, A-C-E, that is angiotensin converting enzyme. And this enzyme is coming from your lung tissue. So your lungs release ACE. And the ACE now is going to convert angiotensin 1 to angiotensin 2 right here. You'll notice here, angiotensin 2 is shorter than angiotensin 1. So you go from 10 amino acids to 8 amino acids, right? So now angiotensin 2 now is going to start having, if you notice here, a wide scale impact on your body. This guy is going to be responsible for a lot of different things. Well, first, angiotensin 2, is going to trigger your hypothalamus. It triggers your hypothalamus and causes your hypothalamus to make you thirsty. Right? Of course, if you are thirsty, you will want to drink. And when you drink water, that's water that will be added to the blood. That will cause your blood pressure, of course, to rise, which means now there will be enough blood pressure for more plasma to leave the blood, go back to the tissues to make more tissue fluid so your cells can be rehydrated. That is why, of course, you get thirsty. But while that is going on, angiotensin 2 also impacts your heart, your cardiovascular system, right? It causes your heart, well, it causes the blood vessels of the heart the blood vessels to constrict, right? Which causes an elevation in blood pressure. It might also cause your heart to beat more strongly, but this is dependent on how dehydrated you are, right? So you might find your heart beating faster if you're very dehydrated. But that is severe dehydration. But of course, you also cause vasoconstriction. So it affects your heart, it affects your blood vessels. Of course, if you vasoconstrict, means you're squeezing down on the blood in your vessels. That causes your blood pressure to rise. Okay? And remember, blood pressure is important. Without blood pressure, your heart actually will stop. Without blood pressure, there's not enough force carrying blood to your brain, which of course will kill you. There will not be enough blood pressure for your kidneys to function. So you need to maintain your blood pressure within the normal range. So vasoconstriction, when your blood volume is low, helps to raise the pressure and keep you functional, right? And of course, another thing that is done is that angiotensin 2 triggers the adrenal cortex to release aldosterone, right? Aldosterone itself is going to work now on your kidneys. What it does is it causes the reabsorption of sodium, okay? So when it says salt and water retention, it's really sodium. Sodium from the urine, this is what happens at the distal convoluted tubule, will be absorbed and sent back to the blood. Now, you must say to yourself, if you're putting all of this sodium in the blood, aren't you making the blood increasing the osmotic concentration of the blood? That doesn't seem like a sensible thing to do. But when you're adding sodium to the blood, yes, you increase the osmotic concentration. And why do you want to do that? Because of water. As the sodium is flowing from the urine into the blood, water from the urine and from the kidneys themselves will flow into the blood behind that sodium. So you're taking water from the urine in the kidneys and you're sending it back to the blood. You're simply using sodium as a means of moving the water. And why don't you just pump the water out of the kidney? Because you do not pump water. I think I mentioned it to you last time. You never pump water under any circumstances. Your body doesn't do that. Water is heavy. It's hard to move. It's incompressible. It's a pain in the butt. So if you want water to move, you make water move in its own devices. You make water diffuse, move by osmosis, etc. And water, of course, follows salt. Wherever there's plenty of salt, water will follow, right? So if you put the sodium in the blood, water will leave the urine and follow the sodium. Simple as that. Right? It's the same reason, of course, why when you sprinkle salt on a slug, it kills the slug. All right? The salt on a slug forms a very high, osmotically high concentration fluid on the slug, and it pulls water from the slug cells, dehydrates the slug, kills the slug. Right? Anyway, so that is angiotensin 2 for you, right? So the renin-angiotensin-aldosterone system will result, of course, by the three mechanisms I just mentioned in the increase in your blood pressure and hopefully your blood volume, assuming you're drinking water, right? All right, so that, of course, is what this is saying here. Any questions so far? All is well?
Sir,
Yes.
is that the same effect as saline in the body?
What kind of saline?
Salt, water.
Yes, but normal saline, hypertonic saline, hypotonic saline, it makes a difference, right?
Okay.
When we do electrolytes, we'll talk more about that. But depending, all right. When somebody's severely dehydrated and you carry them to the hospital, you get a saline drip, right? But that saline is what we call hypotonic saline. The idea here is that there's salt in there because it's saline solution, but there's more water in that saline solution than there would be in your normal blood. So you're adding water to the blood and a little bit of salt, right? You never want to add pure water to your blood because it can cause a serious drop in your blood osmotic pressure, which can actually kill you, right? So somebody who's severely dehydrated, if you just give them straight water, it can actually kill them, which sounds weird. Give somebody water to drink and they're dying of thirst and it kill them, but yes, it can, right? When we do electrolytes, we'll talk more about it, right? But remind me about that question when we reach there. All right. Now, as it says here, we're talking about peritubular capillaries now. Remember what the peritubular capillary is. The efferent arteriole, when it leaves the glomerulus, a branch of it comes off and wraps around the nephron. And that branch is called the peritubular capillary. And it is the peritubular capillary where most of the reabsorbed substances from the nephron will go to get back into the blood, right? So the peritubular capillary is important for reabsorption of substances from the blood, right? And remember, it is for the cortical nephrons. You're not going to find them in the juxtamedullary nephrons, which I haven't spoken about very much yet, but we'll get to that, right? So as it says here now, your blood has unusually high colloidal osmotic pressure here, and the BHP is only 8 millimeters of mercury. What does this mean? It means that the force pulling substances into the blood, remember that's the colloidal osmotic pressure, and holding substances in the blood is high, right? So the peritubular capillary wants things. It wants to pull them into the blood because of this high COP. And the blood hydrostatic pressure, which is normally 60, remember, in the glomerulus, is now 8. Which means, of course, the force pushing substances out of the blood is very, very low. So what happens? As the peritubular capillary is wrapped around the nephrons, as the nephron is pulling substances out of the urine and dumping them outside of itself, near the peritubular capillaries, this stuff goes right back into the capillaries. So it goes back into the blood. So when you pull salt out of the blood, out of the urine at the loop of Henle, and you dump the salt in the interstitial fluid, it goes right into peritubular capillaries. When you pull glucose out at the PCT, it goes right back into the peritubular capillaries, right? So because of this setup here, high COP, low BHP, the peritubular capillaries can pick up what the nephron is putting out and sending back to the blood, right? It also mentions that water absorbed by osmosis and carries other solutes with it. So the idea here now is as you're making the blood more concentrated in the peritubular capillaries, by putting in the glucose, by putting in the salt, these things, the osmotic pressure increases. And of course, what they would say earlier, water follows salt. So the water itself now, it will leave the urine and flow into the peritubular capillaries following the solvents, right? And whatever is dissolved in the water gets carried along as well, which is what they mean by solvent drag. And by doing this, you're reabsorbing water, you're reabsorbing salt, you're reabsorbing hormones, etc., etc., etc. And all of that stuff goes right back into the blood. Remember, you have to reabsorb 99% of your filtrate. So the peritubular capillaries are very important for this process, okay? All right. So we already know what the PCT does, but we're going to talk about it in a little bit more detail than we did before, okay? You already know from CSEC basically what the PCT does, right? You know, those of you who remember CSEC. Now, as it says here, the PCT reabsorbs 65% of the glomerular filtrate to the peritubular capillaries. So 65% of what is filtered out at the glomerulus returns to the peritubular capillaries at the PCT, which means by far the PCT reabsorbs the most filtrate and sends back to the blood, okay? It's the most important part of reabsorption. Now, it mentions here that the PCT is long, great length, and prominent microvilli and abundant mitochondria. Simply put, this is what makes the brush border. So the one of the reasons why the PCT is the proximal convoluted tubule. Remember I said to you a convolution basically means twist up, twist up. So the PCT, because of all the convolutions here, it's all wrapped up. You take a long tube and you basically twist it up so it will fit, right? In the space for the nephron. But the tube itself is actually very long if you stretch it out. But of course, it's wrapped up, so it'll fit. And then around this PCT now, you have the peritubular capillary, right? And as you pull things out of the PCT by osmosis, by simple diffusion, right? I remember osmosis is a special form of diffusion. Everybody should know that, right? And osmosis is water movement only, and the water has to move across a cell membrane. If water moves between cells, that is not osmosis. It has to move through the cell to be osmosis, okay? And you have simple diffusion. You all know what facilitated diffusion is, I assume. And then you have, of course, pinocytosis, phagocytosis, right? And you have exocytosis, all of these things, which you should hopefully have remembered from CSEC. But we'll mention them as we go along, right? So by all the means I just mentioned there, the PCT takes substances from the filtrate, pushes it outside, and from there, it gets reabsorbed into your peritubular capillaries, right? So as it says, and of course, active transport requires ATP, which of course is why you need a lot of mitochondria in your PCT cells. The microvilli, and everybody knows what microvilli are for, they increase surface area. So the microvilli increase surface area in the PCT. So of course, pulling substances out of the filtrate is more efficient, right? Now it says reabsorbs a great variety of chemicals than any other part of the nephron. So of course, as you know, the loop of Henle is concentrating on salt and water. The DCT is pulls out a little bit of water, but its job mainly is secretion, which is adding stuff, not taking it out. The collecting duct is mainly as well for the absorption of water. So the PCT absorbs the largest variety of substances, right? Mentions here that it does this by the transcellular route, which means through the cells, and paracellular route, which means between the cells, right? So either the substances can squeeze between the PCT cells if they're small enough, or they have to be passed through the cells themselves to leave the tubule, okay? All right. So far, so good. Now, here's an important term, transport maximum. Transport maximum is essentially looking at how high the volume of a substance has to be in the blood before it starts showing up in the urine. Right? So basically, if the volume of a substance is below transport maximum, it should not show up in your urine. So glucose, of course, is the main one we use in transport maximum. If the glucose levels of your blood are below transport maximum, it means that your PCT cells can pull all the glucose out of the urine before it heads to the bladder. So if you do a urine test, you should not get any glucose in your urine, okay? And of course, if the transport maximum is exceeded, the PCT cells can't pull the glucose out fast enough, so some of it ends up in your urine. That is, of course, when you look at a positive test for glucose in the urine, right? It mentions here that for glucose, transport maximum is about 220 milligrams per deciliter, right? So anything more than that, you'll get glucose in your urine, which of course is one of the first, or what we consider to be a major sign of diabetes, right? Diabetes mellitus, anyway. That said, I want you to understand something. There are times when you're going to have glucose in your urine and you're perfectly fine, right? For example, when you eat ice cream, there will be glucose in your urine. If you eat a banana and cheese, there will be glucose in your urine. You see where I'm going with this? Anytime you consume a reasonable amount of a substance with plenty of simple sugars in there, they're going to end up in your urine because your blood glucose levels going to spike like crazy. And there's no way your PCT can pull all of that glucose out of the urine before it passes through. Now, this is one of the main reasons why when you're doing an actual glucose test for urine glucose test, you can't just stroll into the doctor's office after lunch and take the test, because your blood glucose level will be elevated. You can't drink no soda or anything like that. There's a reason why we call the blood glucose test, sorry, not blood glucose, the urine glucose test, a fasting test. Well, the blood glucose one is also fasting, right? So the idea here is that you're not supposed to eat anything or drink anything with any sugar in there or carbohydrates for 12 hours. Why? So that your glucose levels have dropped to their minimum. Okay? So therefore, if they're at their minimum acceptable level, no glucose should appear in your urine, assuming you're healthy. So after 12 hours, you draw the urine, and it's better to do the overnight one. So that what they normally tell you to do is that from 9 o'clock the night before, you will not eat any food, you not drink anything but water. Some of them tell you don't even drink water. Right? But if I'm honest, you can probably get away with drinking water. As long as you don't overdo it. Now, generally speaking, you go in the next morning, normally by 9, 10 o'clock, they'll take some urine or draw some blood, and then they'll test for glucose concentration. And of course, if you're doing the urine test, your blood glucose, well, your urine glucose should be 0. Right? Now, that said, I can tell you that in Jamaica, you will go to the doctor's office, them say, hey, come in 10 o'clock tomorrow morning, 9 o'clock tomorrow morning is your appointment. Don't eat anything from the night before. You go in there, 9 o'clock, then it's 10 o'clock, then it's 11 o'clock, then it's 3 o'clock, and I'll know nobody will come to you. So you have to be aware that if you ever ask, unless you're doing it privately, just be mindful that you might be fasting for up to 24 hours, if I'm honest. Because I know people it has happened to, right? Anyway, let's now examine this lovely diagram here. What we're looking at here is a section through our PCT, right? So we have over here we have the lumen. So this is where the filtrate is flowing, right? We have our PCT cells here. And we have our peritubular capillary over here, okay? Now, if you notice here, this is showing all the different types of transport occurring at the PCT that will allow the PCT to absorb that 65% of filtrate, right? Now, where you see active transport, it means, of course, that ATP is being used to pull substances through the cells and then dump them into the space here between your PCT and your peritubular capillary, okay? Facilitated diffusion is diffusion through protein channels that does not require any ATP, and it only allows specific substances through. So for example, you can have sodium channels that are facilitated diffusion. Only sodium can move through these channels. But you must always have a higher concentration of sodium out here and a lower one inside, and that will cause the sodium to flow through all the way to the next side. Remember, facilitated diffusion is still diffusion, just through proteins. So you're still going from a high concentration to low concentration, just through proteins, right? So we also have a system where we could have what we call an antiport system. The idea here is that you're swapping out. You're taking one substance in, and at the same time, you're replacing it with another substance, right? So you're swapping out. So for example, you could be passing protons out, right? So H plus could be going out. At the same time, you're taking in sodium, right? That's an anti-port system. So one goes out, one goes in. It's normally protons or bicarbonate ions, right? So if you see anti-port, remember one substance is swapped for another substance. Active transport, you're moving substances against the concentration gradient into your PCT cells. Facilitated diffusion, you're moving substances by diffusion through protein channels into your cell, right? And specific substances, right? Some substances are small enough that they can, and they have the right charge, they can just diffuse right through the cell membrane. You don't need anything special for them to move through, right? So fat-based substances, for example, can diffuse right through cell membranes, no problem. So some hormones, for example, like testosterone, can just diffuse right through, no problem, right? Now, in terms of paracellular, sorry. In terms of paracellular now, remember, it runs in between the cells. So it mentions your water, for example, can move via the paracellular route. It just runs in between the cells But remember, water, it needs incentive to move. So when you're pumping out the sodium, right, and the chloride ions, and to some extent the glucose and the amino acids, you're moving all of these things into the fluid right here, so in front of the peritubular capillary. So this fluid becomes very concentrated, high osmotic pressure. So water, of course, happily moves along between the cells and moves into that fluid, because water goes from a solution. Well, how you go? If I say water moves from a hypotonic solution to a hypertonic solution, I'm hoping you all understand what that means. You can let me know if you don't, right? But basically, water moves from where there's plenty of water to where there's less water in comparison and more substances dissolved, right? Now, as the water is moving, other substances like urea, which you're trying to get rid of, uric acid, which you're trying to get rid of, and some sodium will also follow behind the water, right? So even though you're trying to get rid of the urea, some of the urea still leaves the PCT and ends up going back into the blood eventually, right? Because you can't stop the urea. The urea can diffuse right back through. No way to stop it. So every time blood passes through the nephrons, you're basically only losing half of the urea because diffusion goes from high to low, which means as long as there's more urea in the fluid here than in the blood, urea will always diffuse right back into the blood until it's 50-50, right? So you lose half of the urea only. You can't help it, right? All right. It also mentions things like vitamins can also be taken in, active transport as well, and then passed once again back to the blood, all right? Even lactic acid, which technically you're trying to get rid of, can also pass through as well, okay? All right. So hopefully this makes sense to you, right? So remember the mechanisms that can be used to move substances from the lumen into the blood via the peritubular capillaries. All right. Still no questions, so we keep going. So, tubular secretion of the PCT and the nephron loop. So we have waste removal at the PCT. So what are we removing as waste? Urea, only about half of it. Uric acid, same thing. We're also getting rid of some bile salts, ammonia, catecholamines, and which of course is a drug, and other types of drug will be removed from the blood at the PCT, right? As long as these substances do not diffuse easily through the cell, they will pretty much stay in the urine, right? Now, the PCT also helps with acid-base balance, right? I mentioned to you already that in the antiport system, you can actually pass protons out of the cells into the lumen and replace them with something like sodium. So if you have a high concentration of protons in your tissues, they end up in the fluid and then they diffuse into the PCT cells and then they can be swapped out with sodium. So you can actually reduce the acidity of your interstitial fluid by putting the acid causing, let's call it that, protons into the lumen and retaining sodium instead. So you're dumping out the acid and picking up the sodium. Therefore, you can adjust the acidity of your blood and your tissues, right? If your tissues are too alkaline, there's too many bicarbonate ions there, then you can do the opposite. You can absorb protons from the urine, send that back to the blood, all right, and swap out sodium for it instead. So you can either add protons as you need or take them out as you need at the PCT to balance your pH, right? So that is where acid-base balance comes in. When we do electrolytes, we'll talk more about that, right? Now, of course, from the PCT, we move on to the loop of Henle. You already know loop of Henle As we said, two major things that happen here, water and salt reabsorption, right? And of course, now we'll talk about loop of Henning in a little bit, but DCT and collecting duct. Remember what we said about the DCT. The DCT is where you have secretion occurring, right? Remember, filtration at your glomerulus, renal corpus, really. Absorption at the PCT, absorption at the loop of Henle, and secretion at the DCT. The DCT, you're taking substances that are in the blood that are usually poisons, but are in low concentration or are too big to filter out easily from the blood, and then you're forcing them into the urine. That is the secretion part. You're actively pulling them out and dumping them in the urine, okay? So secretion. Now, it mentions here the effects of aldosterone. So it says when the blood pressure drops, remember we just spoke about it, angiotensin 2 formation. Remember, angiotensin 2 is coming from your liver. It stimulates adrenal cortex. Adrenal cortex secretes aldosterone. Aldosterone promotes sodium reabsorption. Remember, we went through this already, right? So we're not going to bother with that again. I do want to mention ANF, atrial natriuretic factor. Because normally when we talk about blood pressure, we're talking about causing your blood pressure to rise, okay? But you also have a mechanism to stop your blood pressure from getting too high. This is now your ANF. So if your blood pressure gets too high now, you stimulate the right atrium of all places of your heart to release ANF. Atrial, which of course is why it's called atrial, right atrium, natriuretic factor, and that promotes sodium and water excretion, which now means that at your DCT, instead of sodium being absorbed and sent back to the blood, you're pulling sodium from the blood, dumping it in the urine. And remember, once again, water follows salt. So as you're moving the sodium from the blood into the urine of the nephrons, water also leaves the blood, goes into the nephrons, so your blood pressure, of course, is going to drop, right? That should be okay, right? All right. Then now we have ADH. ADH is one of these things once again, I know I'm sound like a broken record, you'd have done at CSEC, right? But please be aware, ADH is also called, and this is the name we normally use, vasopressin. All right? ADH is also known as vasopressin. As a matter of fact, you might see vasopressin in your test instead of ADH. You might see ADH, you might see both, because it's the same thing, right? Please be aware, vasopressin, ADH, same thing. Now, ADH is released now when you have dehydration, okay? So, as you know, your hypothalamus monitors your osmotic concentration of the blood. When your blood becomes too concentrated and your blood pressure starts to drop, your hypothalamus picks that up. It then causes the release of vasopressin / ADH from your posterior pituitary. ADH / vasopressin goes into the blood, circulates to the kidneys, goes into the filtrate, right? Ends up in the collecting duct of the nephron, right? Once it gets to the collecting duct of the nephron, the collecting duct is normally impermeable to water, as in water can't leave at all water can't enter, nothing happens. But ADH will bind and form structures called aquaporins. So aqua, as in water, and porin, as in pore or hole, right? So it literally makes water holes, right? So the aquaporins, which are formed with the ADH, allows water to be pulled from the urine and go back into interstitial fluid, right? And from there, this water can make its way back to the blood. So ADH causes you to pull water from the urine and essentially keep that water for the blood. So if you're getting dehydrated, you release ADH, okay? All right. This is showing what is this is showing you here 300, 600, 900, 1200. This is the osmolarity within your kidneys. The idea here is that the deeper down into the kidneys medulla you go, the saltier it is. And this is important, okay? Because if urine is traveling down the collecting duct and you're trying to reabsorb water from the urine, as you're losing water at an osmolarity of 600, in order for you to pull out more water further down, you have to have a higher concentration of fluid on the outside, right? So the higher the concentration of the fluid is that the urine is passing through in the collecting duct, the more water gets pulled out of that urine. Which is why, of course, as the collecting duct goes down the medulla, the concentration gets higher and higher and higher to pull more water out, okay? So that's where you go from 300 in the cortex all the way down to 1200 deep in the medulla. So remember now, vasopressin comes to the collecting duct, forms the aquaporins in the collecting duct. As urine travels down the collecting duct, the aquaporins allow water to leave the collecting duct and go back into the kidney's tissues to be reabsorbed by the blood. The deeper down you go in the medulla, the saltier it is, essentially. So the more water you pull out of the urine. So because of that, you can make very, very concentrated urine, right? So as it says here, osmolarity is 4 times as high deep in the medulla. This is 4 times more concentrated. So you pull out 4 times more water, essentially, right? So as it says, medullary portion of the collecting duct is permeable to water, but not salt. So you're pulling water out with, remember, with the ADH only, and you're leaving the salt in the urine, okay? Okay. So far, so good, I hope. Let's continue. We're near the break, but I want to finish this tonight. All right, I'm just going to skip the break, and I am simply going to continue until we finish, right? Actually, we'll probably finish before 9 o'clock. Control of water loss. So your body can produce either hypotonic urine. So hypo, of course, is less, right? And tonic is referring to the osmotic concentration, right? So in a hypotonic urine, you have less salt, more water, okay? So hypo, less salt, more water, right? So tonic. In a hypertonic solution, or urine in this case, you have more salt and less water, okay? So hypotonic, hypertonic, and of course, if you have equal volumes of both, you are isotonic, okay? So if you're equally balanced, you're isotonic. You have more salt in comparison than water, you're hypertonic. If you have more water in comparison to salt, you are hypotonic, right? This is the urine in comparison to your tissues. All right. So as it says here now, producing hypotonic urine, sodium chloride is reabsorbed by the cortical collecting duct and water remains in the urine. So what does this mean? To make hypotonic urine, you do not produce the ADH or the slash vasopressin. You produce only a small amount of it. So what does that mean? It means the water stays in the urine, you're not reabsorbing it. You reabsorb some of the salt from the upper part of the collecting duct, which is the cortical part, but the lower parts, you don't absorb salt from them. And without ADH, you don't absorb water from them. So you have slightly less salt and a lot more water in the urine. Your urine is now hypotonic, right? Now, if you're going to produce a hypertonic urine, on the other hand, usually your glomerular filtration rate has to fall and you have increased tubular reabsorption. So you're becoming once again, you're becoming dehydrated. Your blood pressure is starting to drop because you have less water in there. So that means that your GFR drops because of that as well, right? So that reduces tubular reabsorption. Sorry, tubular reabsorption increases and filtration decreases. So you're actually producing less filtrate. So you're losing less water, right? And of course, ADH works on the collecting duct as well, which means even more water is being pulled out of the less urine you just produced, which concentrates the salts and other dissolved substances in the water that is left in the urine. Your urine now becomes hypertonic, right? So you need ADH to make hypertonic urine. All right. Now, let's talk about the counter current exchange system. I'm going to do a little diagram for this one. Hopefully, it will simplify things. The counter current exchange system, first of all, this is now for your juxtamedullary nephrons. Remember the juxtamedullary ones? Those are the ones that start out most of the juxtamedullary nephron, remember, is inside the medulla of the kidneys, right? And they have very, very long loops of Henle. And as I said to you, these guys are designed specifically to reabsorb water. Okay? So the counter current exchange system is for juxtamedullary nephrons. So we're going to have, of course, our loop of Henle. Since we're pulling the juxtamedullary ones, are going to have a nice long one. Longer than this, but we'll work with this. Wait. It should be thicker up here. That's our loop of Henle. Right? Now, of course, we know we have sections, so we have descending loop over here. We have ascending loop over here. Right? And of course, we have our thick loop. And our thin loop. Okay? So those are the important parts of the loop of Henle we need to worry about. Now, in order for this to work, we also need a blood vessel, a capillary. We have a special capillary here running beside, well, actually it's wrapped around the loop of Henle, but it makes it simpler to just draw it running beside it. So remember, this thing should really be wrapped around, but I make life more difficult for ourselves, right? So we just draw it running right beside it. Now this vessel here is special. This one is coming from the peritubular capillary. It's called the vasa recta. Okay, the vasa recta, special capillary. Okay. Now, I am going to add some numbers here. So 300, 600, 900, and 1200. This is milliosmoles, M-O-S-M. So this is the osmolarity here. Okay. So here we have cortex. And here we have medulla. Just like we showed before, right? So the idea here is that as you go down into the medulla from the cortex, the concentration or the osmolarity gets higher and higher and higher, which of course means the fluid around the nephron gets more and more concentrated, okay? There's more and more salt on the outside. All right. So that's our setup, okay? So see if you can follow me now. As urine flows down the descending loop of Henle, that's our urine, okay? We're going to have salt. We're going to use the green lines as salt, leaving at the thick loop. I'll say salt, but it's mostly sodium. All right? Sodium and chloride. Leaving the thick loop. Remember, the thick loop is active transport, right? So that's active salt transport. So the salt is being pumped out at the thick loop. Obviously not all of it, right? But that is one of the things that helps to make the fluid around the nephron more salty, right? Now, once you move from the thick loop, and you can see the thick loop on the descending side is not very long, you move now into the thin loop. Let me just draw in some more of these real quick. Because of course, at the other thick loop, you're also going to have salt being pumped out, okay? Now also, because this is urine, you're going to have water as well. So just to remind everybody, the green arrow is salt, mainly sodium and chloride. So of course, the blue arrows are going to be water, right? No water leaves the thick parts. That said, the thin parts are permeable to water. So the thin parts, you will have water leaving. And the deeper down you go, the more concentrated it is, remember. So the more water gets pulled out of the urine as the urine runs down the thick part of the loop. Okay? So, with that in mind, see if this makes sense to you. As your urine travels down the loop of Henle, the urine becomes more concentrated because you're losing plenty of water from the thin loop, but you're retaining the salt. So you're losing water, you're keeping the exact same amount of salt. because salt can't leave the thin part, right? Now, once you start going up the ascending part now, once you leave the thin part, you're no longer losing water. So, once you leave here, now you go into the thick part. You notice the thick part here on the right side is longer than on the left side, right? So, this long bit here now of thick loop, you'll have salt being pumped out, but water stays in, right? Which means as you go up the ascending loop of Henle, the urine is becoming less concentrated. So remember now, going down the loop of Henle, urine becomes more concentrated, more salt is in there. Sorry, the same amount of salt is in there, but less water is in there. As you go up the loop of Henle, you're keeping your water, but you're losing your salt. So the urine becomes less concentrated. Okay? All right. Hopefully so far so good. Now, how does the vasa recta figure into this? The vasa recta itself, the concentration of salt in the vasa recta is fairly low. It starts out at 300 milliosmoles, right? And because the concentration of salt is low in the vasa recta and the concentration of salt in the fluid around here is high, salt leaves the fluid and flows into the blood, right? So that means, of course, that the vasa recta starts picking up salt. And the more concentrated the area is, the more salt the vasa recta picks up. So the blood in the vasa recta is becoming saltier and saltier and saltier, right? So the urine, which is going up at this time, is becoming less salty, whereas the vasa recta, which is going down, so remember the blood is flowing this way, all right? So that's blood. Whereas the urine is going in the opposite direction, right? So as the urine goes up, the vasa recta collects salt from the nephron loop, right? And makes the blood more and more salty. Now, this very, very salty blood then starts to move up now the loop of Henle. So when the descending loop is coming down, of course, the vasa recta is going up. Now, of course, what is outside here? Plenty of water. Because we've been losing water to the fluid, right? And of course, if your blood here is extra salty, the water here naturally wants to flow towards salt because water follows salt. So, what do we have here? We have the vasa recta collecting salt on the way down the nephron loop and collecting water on the way up the nephron loop. So this juxtamedullary nephron preserves both water, or should we say collects more water and more salt than a regular nephron because of this system here, the counter-current exchange system. And it's called counter-current because blood goes in one direction and then urine goes in the opposite direction. So they're moving counter to each other. All right, any questions here? All right. All is well. Let's move on with life. Okay, so that is the counter current exchange system for you, right? So this, of course, is what I have written here. Reread it. It should make sense to you now if it didn't make sense before, right? So here we have again, here is our vasa recta over here, right? Here we have our loop of Henle. You can see the thick loop here on the left, on the right side is much longer than the thick loop on this side. As you're going down, you can see water leaving, but not salt. When you're going up, you can see the salt being pumped out, but not water, right? All right. So this, of course, is a summary of the substances that are being reabsorbed. As you can see, all of these things here are reabsorbed at the PCT, right? And at the DCT over here, some you can see what is being reabsorbed here. Some water, some bicarbonate. I mentioned bicarbonates and sodium already too. Remember, that is acid base. If your blood is too acidic, you need to reabsorb bicarbonates and send it back to the blood. You swap it out with protons. Remember the antiport system I showed you in the PCT? You still have one in the DCT as well. So if your blood is too acidic, you pump out the protons and pump in sodium to replace it. You also reabsorb bicarbonate ions and send it back to the blood as well, all of which cause your blood pH to rise, right? And of course, you also have protons, as we mentioned before, moving into our DCT and eventually ending up in the collecting duct, right? So this is a good little diagram just to remind yourself as to how things are moving. For example, here's our nephron loop again. We have water leaving the thin part, salts leaving the thick part, as we said before, right? All right. Now, let's talk more generally now about urine. We have here now some characteristics that urine is supposed to have, right? Yes, I am. Yeah. Yeah, sorry about that. First of all, what is the color of normal urine? I mean, you all been urinating all your lives. You should have some idea as to what is normal, right? You're expecting urine to be almost colorless, as in very, very lightly yellow. That's normal. If your urine looks like a glass of water, that is not normal, right? It should be very pale, yes, but there should be some color in it. If your urine is literally like a glass of water, you're overhydrated. I need to cut back on some water. I mean, realistically, your kidneys can manage overhydration as long as you don't overdo it, right? So brief periods of overhydration are fine. You know, let's say you got really hot and sweaty, you lost a lot of fluid, and you just drank way too much water at one sitting, you know, like 1.5 liters of water one time, which is a lot of water. Your urine might come out very, very pale yellow, to the point where it looks basically like water. As long as it later on in the day, it returns to its regular color, that's fine, right? If you're dehydrated, your urine can go all the way down to a deep amber color. And amber is sort of a yellow-brown color, right? As it mentions here, it is due to the concentration of urochrome in your urine. So the more and the urochrome comes from the breakdown of hemoglobin, right? So the more urochrome you have in your urine relative to water, the darker your urine will look. So low volumes of urine tend to be darker in color because of that. Now here's the thing. Urine, when it first comes out of your body, is not supposed to have a smell. If you pee and you smell something, you might want to look into that, right? That said, I will put one caveat to that. If you consume a lot of protein, you might find that your urine might have a slight smell to it. That is because very, very high protein diets increase the amount of urea and uric acid you end up with in your urine. So when you're peeing, you might get a little whiff of that. Otherwise, your urine is not really supposed to smell, okay? Of course, stale urine will smell because the urea is going to be broken down by bacteria to ammonia. And of course, you get the ammonia smell from the stale urine, right? Specific gravity. Specific gravity, of course, is the comparison of the concentration of a substance to water, right? To water. So if something is heavier than water, it will have a specific gravity of more than 1, because water is considered to have a specific gravity of 1. Urine is always heavier than water, right? Because urine has stuff in it. And what, by the way, when I say water, of course, I mean 100% pure water. It's supposed to have less, it's supposed to have a higher specific gravity than that. That said, I have a range here of 1.00 to 1.035. 1.00 does not mean it is the same as pure water. It just means that we're only measuring to the 3rd decimal place. If you check the 4th decimal place, you would find something in it, right? So extremely dilute urine will have a specific gravity of 1.00, which is fine, right? Very, very concentrated urine, you're looking at 1.035. That is urine that comes out very amber, looks almost brown, right? Now, as it mentions your osmolarity, which is now looking at blood concentration. Normal blood concentration is about 300 milliosmoles, right? But your urine can actually be much less concentrated than your blood. So your urine can have 50 milliosmoles, right? Which is a 1/6 as concentrated as your blood, all the way up to 1200 milliosmoles, which is extremely dehydrated, right? Y'all must have heard of stories of people being lost at sea or something, or lost in the desert, and you're told, oh, drink your urine, you'll be fine. Here's the thing. If you get lost, per se, and you were properly hydrated before you got lost, and you collect the urine shortly after you got lost, that urine will usually have a concentration that is less than your blood. So there will be more water in that urine than there is in your blood. If you drink that urine, you can actually extract some water from that urine. So the very first urination after you realize you lost, I mean, how many people are going to remember to collect that one, right? You can get away with that. Listen, it's your urine. All you're doing is putting it back where it come from. Not that big a deal, right? However, you cannot drink the second urine or anything after that, because the next time around, your body's going through dehydration, which means you're producing extremely concentrated urine. And if you drink that urine, what you're doing is putting salt back into the blood without any real additional water. So you're actually going to make your dehydration worse. This is also the reason you can't drink salt water, because the osmolarity of salt water is 400 and odd. 500 and odd. Well, actually, yeah, 45, I can't remember exactly what it is. I think it's around 450 milliosmoles, which means salt water is much more concentrated than your blood. If you drink salt water, all you're doing is dehydrating yourself, because you're adding more water than more salt than the relative amount of water you're getting. It will kill you. So if you're lost at sea, you can't do anything else, here what you do. Catch a fish and drink the fish blood. Fish blood is normally around 300 milliosmoles, which is basically what you have. So you can drink the blood of the fish, but you can't drink the water the fish is in, which is kind of weird, right? But the fish, of course, is going to osmoregulate its salt concentration in its blood, right? So just keep that in mind. Drink the blood of the fish. If you can get a hold of a seagull or something, drink the seagull blood too. All of that is fine. Just don't drink the salt water, right? Or your urine after the first time. Anyway, the pH of normal urine is between 4.5 and 8.2, which is a pretty large range. But it's usually around 6. The pH of your urine is heavily influenced by your diet. If you consume a lot of acidic foods, you tend to have acidic urine. So for example, pizza sauce is very acidic. So if you're eating a lot of pizza, a lot of spaghetti and meatballs, them kind of things, you tend to have more acidic urine. If you drink a lot of soda too, you also tend to have more acidic urine because, as you know, soda is acidic, right? On the other hand, if you eat like a lot of ground provisions, yam and cocoa and banana, and even dumpling and these kind of things, they're more alkaline. So your urine will skew more alkaline. Also, veg is almost always alkaline. So like cabbage and carrots and these things are usually alkaline. So if you're like a vegan, you will tend to have a blood pH that is closer, a pH that is closer to 8.2 and to 4.5, right? Which is one of the reasons why UTIs are more prevalent in these extreme vegans, because your urine is so alkaline that it helps to set up a lovely environment in your urinary tract for these bacteria to proliferate, right? Now, urine is supposed to be around 95% water, with about 5% solutes like urea, sodium chloride, and potassium chloride, etc., right? Now, urine, we already mentioned 1 to 2 liters per day, closer to 1 these days than 2, of course, because the hotter it is, the more you sweat, the less urine you're going to make. Simple as that, right? Now, we have conditions called polyuria. Polyuria, you're producing more than 2 liters of urine a day. This is also a sign of diabetes, right? So if you're producing a large quantity of urine, even when the time hot, it's a bad sign. You'll say to yourself, but I'm drinking more water. But normally that is compensated by the extra sweating you're doing. If you have less than 500 mils of urine being produced per day, we call that oliguria. Oliguria could simply mean that you're moderately dehydrated. Okay? Drink more water, it should correct itself. But if you're anuric, as in you're producing less than 100 mils of urine a day, you're severely dehydrated or your kidneys have shut down, which means you're not making any urine at all. Even oliguria might be a sign that your kidneys are failing if you're drinking a normal amount of water. But anuria, assuming you're not dehydrated, is a terrible sign, must be checked out immediately. Nobody is supposed to go 24 hours without producing urine. If that happens to you, get it looked into, right? Now, I mentioned diabetes mellitus earlier. So diabetes, no, remember, this is mellitus. It's associated with polyuria. As I said, you produce more urine than normal, right? This is diabetes type 1 and type 2, right? Type 1, of course, is juvenile diabetes. So normally it is caused by autoimmune system reaction. This normally happens in children where their pancreas gets damaged by their immune system, so they can no longer produce insulin. Type 2 diabetes now is different. In type 2 diabetes, you get what is called insulin insensitivity. You're producing a lot of insulin, unlike type 1 diabetes, but your body does not respond properly to it. So even though you're making plenty of it, you're still not able to remove the glucose from your blood. But in both cases, you will have a lot of glucose in the blood, right? You also have something called gestational diabetes. This occurs in pregnant women. So some women, as this is 1 to 3% of them, when they get pregnant, they get diabetes, right? When they give birth to the baby, diabetes goes away. So they get temporary diabetes. But it's only temporary for some people. Most women who get gestational diabetes will eventually develop full-blown diabetes. So it's almost as if your body is warning you that, hey, something is going on wrong, you need to change your diet. Once you push out the brat, you need to sort something out, right? Then now we have pituitary diabetes, which is caused by hypersecretion of growth hormone, okay? Which once again leads to excessive production of urine, right? All right, so remember, all of these conditions here will result in hyperglycemia, so elevated glucose levels, which are then related to increase in your urine volume. Why? Because the blood is more concentrated. It pulls more water from your tissues into the blood, which increases your blood volume. And as your blood passes through your kidneys, you therefore produce more urine. And because you're dehydrating your tissues, you drink more water. So with diabetes, you're drinking and drinking and drinking and then urinating constantly because of the high level of glucose in the blood, increasing your blood volume, and you're losing that excess volume at the kidneys just to be replaced by you drinking more water, right? Now, if you have no hyperglycemia, right? Wait, why am I talking about diabetes? You know, let's leave diabetes alone. Realistically, that should be for hormones. Instead, let's go to diuretics. A diuretic refers to a substance that increases the production of urine in your body, right? So if you take a diuretic, your urine volume goes up. Now, diuretics are particularly useful in regards to high blood pressure. So if somebody comes in to the doctor's office, they have extremely high blood pressure, and to the point where they say, boy, something is going to rupture anytime now, you give them a diuretic. I think it's also known as a water pill. The idea here is that the diuretic causes an increase in urine production. And of course, the more urine you produce, the more water you lose from the blood, your blood pressure goes down, right? Now, a very common diuretic is one that most of you are very familiar with. And that one, of course, is alcohol. The more alcohol you consume, the more urine you tend to produce. I'm sure everybody's aware of that, right? More alcohol goes in, more urine comes out. So urine is a diuretic. Now, all right. Oh, okay, so ureters. Okay, so we're almost done. So, of course, once the urine is produced, remember the urine passes to the collecting duct first. And then from the collecting duct, it goes down to the renal papilla. And then from there, it enters the renal sinus, which is the space that leads into the minor calyx, right? So collecting duct, renal papilla, renal sinus, minor calyx. Then from there, you go to the major calyx. Then you go to the renal, oh Lord, my brain is I'm blanking. Renal sinus, then to the ureter, and then from there, of course, you go to the bladder, right? So that's the path urine takes to the bladder. So the ureters carry urine to the bladder, essentially, right? Now the bladder itself, the urine and the ureter share a lot of similarities. So these layers are going to be found in each. So you're going to have 3 layers of the ureter. You're going to have the innermost layer, which is a mucosa. You're used to mucosa by now. You have a muscularis layer and the adventitia. So that should sound familiar to you, right? The mucosa layer is made up of transitional epithelium, which means it's good at stretching under force. And I'm noticing here we only have 40 people here, 39 people, so I will not be posting this recording. Now, the muscularis, 2 layers, right? And the adventitia, of course, is the outermost layer. So why transitional epithelium? Because you're going to stretch. As urine is being pushed down the ureters, the ureters have to stretch under the pressure of that urine. And so you need the transitional epithelium to allow it to stretch. As you know from AMP1, transitional epithelium allows for stretching under force, right? And you need the muscle layers for peristalsis, which sounds weird, but your ureters actually do what we call a pulsatile movement. So they contract in a wave. It's like peristalsis, squeezing urine down the ureters into the bladder, right? It mentions here that the lumen is very narrow and easily obstructed. So this is why things like kidney stones, if a kidney stone can leave the kidney and enter the ureters and block up the ureters, right? And all of a sudden, you have a problem because you can't pass urine to the bladder anymore and your kidneys start to swell, okay? So the narrowness of the ureters is a problem, okay? All right. So it mentions here that from the renal ureters, from the renal pelvis, pass dorsal to the bladder and enter it from below. So the ureters go behind the bladder, go down, and then go up into the bladder, right, from below. So they enter the bladder from the back towards the posterior part of the bladder, right? And of course, now your bladder is going to have similar layers to your ureter. So if you watch here, here's the ureter going down behind the bladder, and then you can see it entering the bladder at what we call the urethral openings here and here. So when the urine is being squeezed down the ureter, it's eventually squeezed into the bladder via these urethral openings right here. So the bladder, therefore, gets filled up with urine like a balloon, filling with air. Now, the area where the urethral openings enter helps to form what is called a trigone. So the trigone right here, tri, of course, is in 3. So it has 3 parts here. So there are 3 holes. We have the 2 urethral openings, 1, 2, and we have the exit to the urethra, forming that triangular area here called a trigone, okay? Now, the detrusor muscles are going to contract when it's time to empty the bladder and squeeze the urine out, okay? Now, I don't think I'm going to spend too much time on this aspect of things here. I'll probably touch this in the tutorial. So fundamentally, we're done, except for some little things to do with the bladder, and I'll run through those in the tutorial, right? And I'll run through this in the tutorial as well. So we can stop here for tonight. I know I went a little faster than normal. So I'm instead of pushing it too far, let's just stop here. I'll finish up at the tutorials Thursday, Friday, right? But please make sure you read through the renin-angiotensin stuff here. Just to explain once again how the renin-angiotensin-aldosterone system works. Questions on this can come all the time. So please make sure you're good with it, right? And as I said, I'll speak about this micturition thing and the hemodialysis on Thursday, Friday, right? My voice is starting to go, so I'm going to stop here. So please remember, 11 o'clock tomorrow, 47C1, starting at 11:05 the latest, should be out of there by 12 or near 12, right? Your urinary physiology is online already at Moodle, but you're still doing the urinary anatomy tomorrow, so I still expect to see the 8 o'clock class, but we should be done by 10 o'clock, 12:30 the latest, right? It's only 3 drawings we're doing, right? Any questions before we go?
Yes, sir. I don't see where you posted the circulatory lecture. I wasn't here for that class on YouTube.
Go on the YouTube website and you'll find one. So go on my YouTube webpage and you'll find one. You will also find a respiration one there as well. And a lymphatic one there as well. Any other questions? All right.
Sir, is this a multiple choice?
Yes. All MCQ.
Sir, how many questions is it?
I don't remember, 45 or 50, something like that.
Okay, sir.
Sir,
Mm-hmm.
the lab quiz also includes a lymphatic.
No, man. Lab quiz is just respiration alone. Anatomy and physiology.
Okay. All right. Okay.
We're not doing a lymphatic lab quiz this semester. We just don't have the time for it.
Okay, sir. Thank you.
No problem. All right. Anybody else? All is well? All right. Assuming all is well, I will let you all go. I will see you tomorrow. Listen, the in-course test is not difficult as long as you've reviewed the material I've sent you. All right? I'm not going to give you anything that is not in what I've sent you. That said, do not expect the questions to be, what is this? What is that? This is a university. You're expecting university level questions, which means that you have to be able to interpret the questions and then use the data I've given you to answer the questions appropriately, right? There might be scenario-based questions as the multiple choice. So please keep all of this in mind. It's not just going to be a what is this, what is that, all right? That said, it's not difficult, right? Because of the shortness of the semester, the test one is normally not very difficult because, as you know, we have to cut things down. That said, do me a favor, please. Study, which is good. Also, sleep properly before you come to my exam, now, please. Do not come to school tomorrow 4 hours of sleep and then go to the exam at 11 o'clock in the day. You're going to write foolishness, right? And if I'm perfectly honest with you, I'd rather you go sleep and wake up and study tomorrow morning and study until 2 o'clock tonight. Because you'll do better. A tired brain gives foolishness. Do not stay up all night and study. You need a minimum of 6 hours sleep. 8 would be better. That's my advice. Anyway, folks, let me leave you. You all have a lot of work to do. Yeah.
We have labs tomorrow morning, 8 o'clock, same way.
As usual, you still have anatomy lab to do.
And which one you say we are to do? Which lab is it due by 10:30? Which tutorial?
What? I have no idea what you're talking about.
Which tutorial you say we are to submit by 10:30?
I didn't say anything about no tutorial for no 10:30. First I hear that. Did anybody else hear me say that? I don't remember saying.
Yes, something about 10:30 tomorrow morning.
I said to you that the lab will finish by latest 10:30 tomorrow. The lab, not the tutorial. All right? So 8 o'clock to 10:30, then I have to go and sort out the room for the 11 o'clock, right? Which is why, of course, you're doing the physiology online instead of in person. So that there's time to do tomorrow, right? All right, any other questions? All right, folks, have a good night.